Thank you so much for being here. I really appreciate it. For those that are in the room, you are brave souls. For folks that are online, you might not know it, but here in Newport, it is raining incredibly hard. And so most people in the room had to swim here, and I appreciate your presence. For folks online, also, thank you so much for being here. My name is Cinnamon Moffett. I'm the Associate Director of Marine Research here at Oregon State University's Hatfield Marine Science Center. And as I said, we are in Newport, Oregon. This is a hybrid event, so we have folks online and folks in the room. For those of you that might not know, if you have a question and you're in the room, please just raise your hand. And when we get to that section of the talk tonight, and I'll bring you a mic, or you're welcome to go to the mic stand on that side of the room, so we can make sure that folks online can hear your questions. For folks that are online, if you use the chat function on your Zoom screen, you'll be able to ask any questions to today's speaker. But because we're in a webinar format, your mics, camera, and screen shares have disabled. So please use that chat to be able to interact with us. And if you have any technical issues, you're welcome to use that chat function. Our wonderful volunteer, Roseanne, will help navigate any of those challenges that you might be having. Also, just a quick reminder, we sometimes cannot drink in this room. Sometimes we can. Today we can. But if you spill any of that beautiful red wine, please let Miguel know. We've got a cleanup box so we can get that cleaned up before it stains our floor. So just a little reminder there. But again, thank you everybody for being here. This is going to be our last Science on Tap of 2024. So we're excited to wrap this year up with our speaker for today. I just wanted to let folks know, if you are curious about any of our events, you're welcome to go to the HMSC homepage to our events section, and you can get to a calendar and look at all of the types of things we're presenting, or you can sign up on our sign up sheet over there, and we'll get you on a listserv just for seminar events like this one tonight. But for our speaker today, we are so excited to have Dr. Steve Rumrill here. Dr. Rumrill is the shellfish program leader with the Oregon Department of Fish and Wildlife. He's been in that position since 2010. The shellfish program is part of ODF&W's Marine Resources Program based in Newport, Tillamook, Astoria, and Coos Bay, and they are responsible for monitoring and managing recreational and commercial shellfish and fishing, and shellfish habitat restoration and research all along the entire Oregon coast. Steve is trained as a marine invertebrate zoologist, specializing in conservation of population and shellfish and sustainable habitat management. He has his bachelor's and his master's from the University of California, Santa Cruz, and a PhD in larval ecology and marine and estuarine invertebrates from the University of Alberta. Among many things, Steve is also the research program manager down at South Slough National Estuary Research Reserve for many years before joining ODF&W. So he's been working up and down the Oregon coast for much of his career, and we're so lucky to have Steve here. Steve is also my old boss, and so I'm very excited to have him here. So I'm going to hand this off to Steve. Welcome, Steve. Thank you.
All right. Thank you very much. It's okay? Yeah. All right, I want to start off by really just thanking Cinnamon, Roseanne, and all of you for braving the weather to come on out here and spend this time. I am very excited and happy to tell a story here that builds really off of the great work and great seminar that we had last month from Dr. Julia Parrish, talking about the effects of marine heat waves on the mortality of seabirds. So I'm going to tell a similar story here, but I'm going to talk about shifting nearshore conditions and the consequences for Oregon's marine species of greatest conservation need. I'm looking at my friend Greg here, who is working on that issue right now, and I'll provide some definition on why I'm using that weird word. I understand that lots of people online and that I'm confined to this space. Is that correct? In the floor? All right. So to help me stay in the bounds here, I'm gonna make this space a little bit more familiar. I think that'll help. Like Julia, when you're confined in this space, you feel like you're trapped. And in this case, with Dungeness crab all around me, I guess I feel like I'm a chicken wing or a cockle, some bait here. I'll try to do my best. All right. So with that, I'm going to get started. And how do I advance here? Let's see. Either way. That button? Okay, great. So thanks, Cinnamon, for that introduction. Let me just add to that. So I'm the shellfish program leader within the Oregon Department of Fish and Wildlife. I'm a marine scientist and have been practicing on the coastline here since I was about 20, and had really wonderful mentors that got me going there. The most fun parts of my job, of course, over on the right, doing field work, gathering observations and turning that into a science project. I'm also a fisheries manager, and that really means managing people's activities. And the fun part of that, of course, is sharing stories, talking with people, interacting with people. And I'm also a coastal resource policy advisor. And in that capacity here, working with my colleagues, Scott Groth, Kendall Smith, we take science and translate it into changes in rules, sometimes changes in statutes, change in policy. And it's very satisfying when you have the opportunity to take some good science and make the world better. And so it's been a very satisfying career along those lines. The scope of the shellfish program, we manage the commercial and recreational shellfish fisheries, Dungeness crab, commercial in the bay, not the big ocean commercial crab. That's another part of our program. But we also manage the Oregon pink shrimp commercial fisheries, sea urchins, bay clams, razor clams, and others. On the recreational side, Dungeness crab, red rock crab, abalone, bay clams, razor clams, and others as well. It's a diverse portfolio of marine invertebrates. Now I want to get started with a quick personal anecdote. I'm wearing my grandfather's jacket. It's hot in here. I'm about to take it off. But I thought red and green, kind of the holiday spirit. But what I wanted to do is, as a third generation guy out of Monterey Bay, my grandparents and my mother, right in the middle, that little girl, were raised during a period of abundant abalone. And the anecdote was that there was so much abalone, they were sick of it. My mom would complain to her father, my grandfather, Art Peterson, "Can't we have chicken? Can't we have hamburgers? I'm sick of abalone." The anecdote really was that in my life, I never saw that myself. That was back in the 30s, but that abalone were very, very abundant. And we build from that point. I'm going to take this off now. Thanks. We have to build from what we can gather as scientists, quantitative information, monitoring and measurements, surveys, technical assessments of condition. That's what I'm going to talk with you about. But we also build in anecdotes, personal stories that we hear about things. So the preamble here for this talk really is that nearshore ocean conditions are changing along the West Coast here. And these recent shifts in ocean drivers have contributed to broad-scale impacts to marine and estuarine habitats. And unfortunately, there have been consequences for several of Oregon's marine species of greatest conservation need. Some species have declined, like the abalone. Other species, like the green crab, have expanded greatly. And we'll talk about some of these here as we go. So the big picture is that there are changes to our rocky reef kelp communities and kelp habitat over the past decade, really 2013 to 2014. Areas that have been historically and in our knowledge operated as dense, diverse kelp forests have now transitioned into urchin-dominated areas like the photograph shown on the right. So is this habitat transition permanent or is it temporary? What do we know about it? Who are the players? So the ecological consequences have included a bunch of different species. I'm going to draw a picture, and I'm starting here just with the graphic to show it through, but through this talk, to look at the marine heat wave, the significant marine heat wave of 2014 to 2016, and the prolonged warming associated with successive El Niños, contributing in some way to the increased abundance of purple sea urchins and the transition of kelp habitat in the subtidal into urchin barrens. We have starved and undernourished sea urchins now roaming around, foraging out there. Concurrent with that, we had observed sea star mortality, the mass mortality of multiple species of sea stars back at that same time period, 2013 to 2015, and continuing to the present day. We've also observed a decline, significant decline in the populations of red abalone here in Oregon, to the point where we had to suspend the recreational fishery indefinitely. That's continuing on. And now we're also seeing, of course, a patchy demise of kelp. So we'll talk about all of those different players here. The goal of the talk today is just to provide a summary of recent work by ODFW, the Marine Resources Program, to characterize the current status and historic changes in our subtidal rocky reef communities. So the outline for this talk, I'll talk a little bit about shifting baselines and metrics for change, talk a little bit about the primary ocean drivers, warmer ocean waters and marine heat wave. I'll do some definitions of the Oregon Nearshore Conservation Strategy and talk about these species of greatest conservation need and use that language. And then I'll present data sets from new surveys looking at the ecological consequences for bull kelp, for abalone, sea urchins, and sea stars, and then finish off with a few questions about the future. So the take-home message that I want to give to you, I'll just give to you up front, in that tell them what you're going to tell them, and then tell them again at the end. Here's the take-home. The magnitude of ecological change in Oregon's rocky reef habitat is unprecedented in recorded history. We're really at a precipice of big changes here. Key questions for us. So do the current communities represent a shifting baseline or some type of new normal for our rocky reef habitats? Will the community continue to respond and adapt into some type of alternate stable state into the future? And what about ecological resilience? Is it possible to help enhance abalone, sea stars, and kelp to somehow assist with recovery? So what about shifting baselines? If we think about what are shifting baselines, they're really a metric of change measured against a previous reference point. And it's problematic when we think about shifting baselines. Why is it a problem? Well, important long-term changes can go unrecognized because new generations of scientists, citizen scientists, and naturalists accept the situation in which they were raised as normal, okay? And then really that contributes to a continual erosion of accepted thresholds for environmental conditions. The graphic is just hypothetical for red abalone. In my life, I became aware of red abalone when I was about 18 or so. And so, they were already in great decline. So I suffer from that shifting baseline about what is new, what is normal, when in fact, we don't really know what they were like hundreds of years ago. So we suffer from that shifting baseline based on our own awareness. Shifting baselines got a lot of attention. Daniel Pauly from British Columbia, fisheries scientist there, wrote a very important paper, Anecdotes, coming back to those anecdotes, those personal stories that are not quantifiable, and shifting baseline syndrome in fisheries. Just want to read you this quote. Each generation of fisheries scientists accepts as baseline the stock size and species composition that occurred at the beginning of their career and uses that to evaluate changes. It's dangerous, but we all do it. Taking another example from Paul Dayton, a little more recent, 1998, really focusing in on the kelp habitats in his important paper. And the quote there, patterns and mechanisms observed over 25 years in a large kelp forest suggests that definition of a meaningful benchmark is impossible because many of the large animals have been gone for years to decades, and kelps are sensitive to large-scale, low-frequency El Nino Southern Oscillation events and longer-term regime shifts. Paul knew that a long time ago, and we're still learning that same lesson now up here in the north with bull kelp forests. So what about alternate stable states? What is that? So alternate stable states are different configurations for an ecosystem that can be maintained under the same external conditions, or really it's a transition from one state into another after some kind of major or significant perturbation. And in this case, stable state one being a kelp forest transitioning into a maybe, perhaps, alternate stable state of urchin barrens. What are the major perturbations? It could be marine heat waves, could be episodic recruitment of sea urchins, loss of key predators out of our systems, sea otters and sunflower sea stars, could be others as well. So that's an important concept here as well, alternate stable states. Okay, I'm going to shift over and now talk about really that perturbation, marine heat waves, persistent marine heat waves and ocean warming along the Pacific coast. We know from many talks here, and I'm just going to say it again, that marine heat waves disrupt nearshore marine ecosystems. And the reported impacts include altered primary productivity out in the nearshore waters, proliferation of harmful algal blooms, displacement of habitats, and then what I'm going to talk about here, changes to populations of marine species and disruption of commercial and recreational fisheries. The graphics just show the formation of that huge, significant marine heat wave known as the blob back in 2014, and that graphic on the right showing persistence up through 2022. If you look closely at the graphic image on the right, you can see that thin line, that thin ribbon down along the Oregon coast of cold water, that blue cold water there due to upwelling. And due to the vagaries of wind and upwelling, sometimes that hot water, warm water, washes ashore, other times it doesn't. There's another graphic just showing that as well, a larger situation where there's more cold water off Oregon and Northern California. But you can imagine as that warm water moves in and offshore, we feel that at different places along the coast. These graphics just show a retrospective analysis of sea surface temperature anomalies. And the graph on the top just shows the total surface area of marine heat waves, if you think about it in terms of a big spatial footprint out in the water. And you can see that it bumped along from 82 till 2026 with sometimes warmer, bigger footprint and smaller. Others, significant amount in 1998. But wow, 2013 to 2024, look at that, huge spatial extent of the coastal ocean wrapped up in marine heat waves. NOAA, the Integrated Ecosystem Assessment Blob Tracker, is really the go-to place for me to go and have a look at it. And so I just pulled up the most recent forecast here from November of this year. So we're seeing remnants of a large marine heat wave over the past April to August of 2024, and it decreased in October last month. We're now in current El Nino Southern Oscillation neutral status, and the forecast is for a possible transition to cooler La Nina conditions over the winter 2025. That's some good news. Hope it'll stay like that. Interesting here, if you look at the percent of the different ecoregions on the left, from Washington down to Southern California, where are the marine heat waves really hitting the hardest? And you can see it's in Oregon and Northern and Central California. Interesting graphic there. So we're really feeling that impact of these unusually warm waters here from Central California, Northern California, Oregon. And then finally, if you take a look at just the really close high detail information, in this case, time series measurements of temperature and salinity, other parameters, right at Cape Perpetua Marine Reserve by our program there. You just look at that second one down from the top, seawater temperature back in 2015 records, showing that very unusual high warm water, those three big blobs of it coming ashore here as winds pushed it back. So we had unusually warm seawater temperatures for about a week in July, another week in mid-August, another week back in end of August to September, about 28 days of unusually high temperature water back then. Take a look at that again on a time series, and you can see that significant 2014 to 2016 warming event, followed by this successive prolonged warming with minor heat waves or La Ninas occurring. So we've seen a change in our nearshore oceans. So we can ask this question: how did the marine heat wave or ocean warming impact ecological communities in our shallow rocky reef habitats? And in order for us to address that here within ODFW, we need to ask, what are Oregon's marine species of greatest conservation need? So what are they? Those are species that are designated by state wildlife action plans as species most in need of conservation actions. Again, I'm looking at Greg, as Greg Kretzkowski is working on this right now on the revision, doing a great job. This document is really important for us, and it's updated every 10 years or so. But what are the criteria for species of greatest conservation need? They can be Endangered Species Act threatened or endangered species. They can be species that are suffering low abundance or declining populations, those that have suffered habitat loss or significant threats to disturbance. But they can also, in our case here in Oregon, be ecologically, culturally important, economically important species. So when we worked with our colleagues to put together the Oregon Nearshore Conservation Strategy, that's the nearshore component of the overall Oregon Conservation Strategy. The purpose for the nearshore part is to promote actions that conserve ecological functions and nearshore marine resources to provide long-term benefits to current and future generations of Oregonians. The nearshore strategy here in Oregon has a section that describes the different ecoregions from north, central, and south, identifies conservation issues and priorities, identifies a series of limiting factors and recommended approaches to address them, and then puts together that list of nearshore strategy species. And I think we had 88 in the 2016 version, and then also identifies opportunities for conservation. So it's a diverse group of species at the nearshore level, 6 marine mammals, 10 seabird shorebirds, about 54 fishes, 14 invertebrates, and 4 species of algae and plants, including bull kelp. And for each of these, identified its special needs, limiting factors, data gaps, and conservation actions. So using that framework, that allowed us to put together a grant proposal and ask the U.S. Fish and Wildlife Service for financial support to help us characterize the status. So our grant was titled Filling Critical Data Gaps to Address the Recent Declines in these Species of Greatest Conservation Need, focusing on the urchins, abalone, sunflower sea stars, and the primary kelp habitat along the southern Oregon coast. Our project had 3 objectives. First was to characterize the current status of those priority species with scuba surveys and also advanced underwater imaging with a remotely operated vehicle in shallow and deep water habitats. Our second objective was to determine the location and spatial extent of kelp habitat with high-resolution imagery, aerial images, and contribute to evolution of a kelp monitoring program here in the state. And third, to generate new time series data to measure variability in those key nearshore parameters at the scale that's relevant to these species of greatest conservation need. So here are those species, and they include red abalone, purple and red sea urchins, flat abalone, the sunflower sea star, and bull kelp. So here's the picture again that it started. Those are the characters, and what I'm going to do now is just walk through this cast of characters and show you what we've learned as we've gone ahead. Those characters, if you put that together into a flow diagram here, you can really see some of the working hypotheses about the proximate and distal causes for those situations. We start in the upper left with shifting ocean conditions and marine heat wave. You can see that through these red lines, it's contributed to a decline in kelp, and we're not sure how. So in the white text is just asking some questions. Is that through decreased spore production and growth, or is that really just through grazing pressure from sea urchins? What's the causative for this? The marine heat wave is also associated with an increase in the abundance of sea urchins. Is that through larval production and somehow massive recruitment? We're not sure. What about the marine heat wave impact on sunflower sea stars and other communities of asteroids? We've heard a lot about mass mortality through sea star wasting disease, some good work identifying possibly a virus or disruption of microbial function at the epidermis level there. But the outcome, the observation and the result has really been the decline of sunflower sea stars as well as other species. With the decline there, has that released sea urchins from predation pressure and allowed them to increase their grazing? And then, how has this helped contribute to the decline of abalone? Has that been through competition for food with the abundant sea urchins or other mechanisms? We don't really know. And so I just put that little note on the top right there, post facto assessment. It's difficult as a scientist to not speculate about what might have happened or contributed to these observations. But the real mechanism happened here along the Oregon coast over a decade ago. So we'd really be doing a lot of arm waving, post facto assessment to try to figure that out. It'd be fun to try to figure it out, but we just don't know. Okay, let's look at kelp beds. So kelp beds as we know, provide ecologically important benefits and habitat in the shallow rocky reef areas. Primary production, nutrient cycling, and carbon storage are recognized as important benefits. Kelp also provides complex heterogeneous structure and also hotspots for marine biodiversity and important habitat for seaweeds, invertebrates, fishes, seabirds, marine mammals. And as well, we also recognize that offshore kelp beds buffer exposed coastlines from waves and storms. Bull kelp it's an annual species, and it has roughly a 1-year lifespan. So the big kelp plant is the sporophyte, and the blades produce spores, sorus. Those are microscopic, move down to the bottom, and they grow up, produce a gametophyte stage and goes into sporophyte stage. There's a complex life history. We hardly know anything about the microscopic stages of bull kelp, and really don't know how vulnerable they are to grazing or disturbance. Bull kelps are naturally highly variable along the Oregon coast. The two photographs at the right show the Orford Reef system in 2014 when we had abundant bull kelp, and then just a couple of years later, very little bull kelp. So we've seen substantial reduction at some sites after the marine heat wave in 2014. For example, Orford Reef, Blanco, Humbug, and Brookings. Scott Marion, who's sitting up on the top and also a collaborator on this project, has handled the part of this project to do aerial surveys of the kelp beds and compare the new imagery taken in 2022 and now again just completed in 2024 with what we knew in the past a decade ago. So Scott organized flights with an ocean imaging airplane that flew flight lines up in the Newport region, the Coos Bay region, and the Cape Arago region. What do we learn from that? Scott should step down here and tell you this. But if you compare the total amount of kelp that we had in 2010 with 2022, statewide, we've seen about a 66% reduction in that area from the California border up to Cape Arago. That's substantial, but it's not the same everywhere. The histogram at the bottom shows some areas like the Rogue Reef right in the middle, where there was a slight decrease, and then compare that to Crook Point, where there's a massive decrease, or over at the Orford Reef area or Blanco Reef, where there have been very substantial decreases. So it's been patchy. We had some areas where bull kelp was hanging in there. Here's some areas where it was not, though. So this series of photographs shows you the loss of kelp in the Brookings region, which has suffered about 82% decline in spatial extent over the past 12 years. And here's another photograph showing you the Blanco reef area, which has even suffered more. My estimates were about 91% reduction over that time period. But bull kelp persists at some other sites. And again, the aerial surveys have shown us hardly any change between 2010 and 2022 at Cape Arago. And at that point, 2010, 2022 at Rogue Reef. We're not sure really, or Scott knows, but I don't know yet what the story is for 2024. Now, bull kelp can't really tolerate huge swings in temperature. So it has a thermal tolerance of 5 to 22, but idealized temperature about 10 to 12 degrees centigrade and maximum growth at about 12 degrees. So it is possible that they have some type of localized adaptation to changing thermal regimes. I mentioned before that bull kelp reproduces by producing spores off the sori region of the blades. And these spores develop into microscopic male and female gametophytes. And then the gametophytes join to produce a macroscopic sporophyte that grows up into the kelp. What do we know about the vulnerability of those very small stages to temperature, salinity changes, to marine heat waves, and also to ecological pressures such as grazing? There's been some work done to look at that in terms of how the sporophytes and gametophytes differ in their vulnerability to temperatures. And again, the ideal temperature is about 10 to 12 degrees. But the experiments that were done down at Sonoma State really showed us that warm seawater results in poor success for these very small microscopic stages. And if we just look back at the marine heat wave of 2016, 2014, rising temperatures into the 15-degree range, they were subject to sublethal thermal stress for various periods of time. How significant is that? We're into that post facto world of trying to figure out things after the fact. So moving ahead here, so we've looked at the marine heat wave possible influence and shown you a significant decline in kelp. Let's move on to talk about multiple species of sea stars. So as you know, sea star wasting syndrome impacted multiple species of sea stars along the whole West Coast, coincident with the marine heat wave 2013. And some sea stars have recovered, like ochre sea stars, but others, many of the ecologically important, big, soft-bodied sea stars, have not really recovered. Sunflower sea stars we recognize as important predators that prey upon sea urchins and other organisms in rocky habitats. And new experiments over the last couple of years have shown that big Pycnopodia, big sunflower sea stars, can consume about 2 urchins every 3 days or so. The onset of sea star wasting syndrome was coincident with the marine heat wave. What happened in Oregon? So the mass mortality of sea stars and sunflower stars occurred all the way down their biogeographic range from Alaska down through California. And you can see that decline in Washington, Oregon, and California. Excellent data came from the NOAA trawl surveys that pulled all that together. And what we saw was 100% declines in California and Oregon in those data sets, about 99% decline up in Washington. No sunflower sea stars in almost 700 trawls back in 2016. So the coastwide decline was about 90% from Alaska down to Baja California. And here in Oregon, our records back from 2009 showed them cooking along okay up into about 2014, and then rapid decrease in 2015, 2016. So there was a multi-year decline, really, from 2012 down to 2018, but that big significant drop associated with marine heat wave. The second note on there is that Oregon has really been a hotspot for the observation of juveniles along the West Coast. So that's some good news here. I wanted to just talk a little bit about the reproduction of sunflower sea stars, just so we're all on the same playing field. So the big adults have separate sexes. They're broadcast spawners, males and females broadcast their eggs and sperm freely into sea. Their larvae develop into planktotrophic or feeding larvae that are drifting around in the plankton for anywhere from 45 days, 60 days, maybe even longer. The key points during that early life phase are the delivery of larval supplies, both their density and the physical mechanisms that bring them ashore. We also recognize that important point in that larval life of larval settlement. That's when this planktonic larval stage settles out of the water column and makes permanent contact with the bottom. At that point, they undergo a metamorphosis and change from a planktonic microscopic swimming sea star to the benthic animal, just reorganize their tissues. That's that important point of metamorphosis. And then recruitment, that's a population biology term. That's when we recognize these new juveniles showing up in our observations. All right, let's shift on over there. So let's shift over and talk now about sea urchins. So many of our subtidal areas are now dominated by sea urchins, and in many cases, those areas support little kelp and seaweed. So we've seen a shift from kelp bed to urchin barren, and we recognize that as a complex process. I mentioned at the beginning of the talk, are we seeing alternate stable states, some kind of phase shift? How resilient are these? Can they go back? And let's see how that's going to work. Will this work? I hope so. Yes. Okay, so these are side-by-side videos, underwater videos. Just I'll let that go for 30 seconds or so. And you can see back in 2011, along these same transect lines, that area is characterized by high biodiversity of algae and invertebrates, lots of algal biomass. Several sea stars would pop up if I let the video run, and a few sea urchins. On the right side, 2019, characterized by low biodiversity, very little algal biomass, very few sea stars, and lots of sea urchins. So we've moved really from what we consider as normal habitat to these urchin barrens. Okay, This got a lot of press, of course, and so when we first started recognizing this, our surveys in 2019 showed about 350 million new purple urchins out at Port Orford Reef alone. That was big news, and it was reported incorrectly that an explosion of purple urchins off the California coast decimated bull kelp at an unprecedented rate, and the creatures spread to Oregon as if they've walked up the coastline. They don't, of course. That's why I wanted to talk about the planktonic delivery, settlement, and recruitment. So they recruited here naturally. We also asked that question at that time, is release from predation by sea stars responsible for this dramatic increase in purple urchins? Our data sets, historic data sets generated by ODFW, have shown some species of echinoderms showed that decrease in abundance, the sunflower sea stars, but also the big giant pink sea stars, Pysastra brevis spinus, the Solaster group, some other big soft-bodied predatory stars. Others showed little change, like the ochre stars or the Henricia blood stars. And then others, like the red and purple urchins, showed increase in abundance, as did the leather star. What do we know about the reproductive biology of purple urchins? Like the sunflower stars, they have separate sexes, males and females spawn, broadcast spawn out into the water. Like the sunflower stars, they have planktotrophic feeding development and a very similar planktonic period of about 40 to 60 days. Those same key points in their life, delivering them to the bottom. Just wanted to point out here from these beautiful photographs, one of my favorite larval forms, the Pluteus. They start off as an early Pluteus, develop 4 arms for swimming and catching food, 8 arms, and then a juvenile rudiment develops on the side. And then when they complete settlement and metamorphose, they just lyse and break down all of that tissue and transform themselves into that post-larval tiny sea urchin. What do we know about those? Their ecology, practically zip. Okay, these histograms show some changes in the abundance of echinoderms in our subtidal habitats going back many years. So I already told you the story on the top, sunflower sea stars declining. But what we've seen is an increase in the abundance of red sea urchins, and going backwards in time, trying to figure out just when did larval settlement take place in that post facto mode. Okay, we know when we saw recruitment in 2015, but that's not instantly coincident with larval settlement. Maybe back in 2012 or so. The purple urchins even a little bit later. So we saw them increase dramatically at 2016 to 2019, and settlement perhaps happening maybe in 2014 or so. So here's a longer time history of ODFW's monitoring of these populations along the Oregon coast here from 1984. That's really the beginning of the commercial sea urchin fishery here on the coast. And the numbers declined and were pretty stable up until about 2014, and then this dramatic increase in 2016, 2019. Our latest surveys done this summer have shown a decrease in the abundance, 2024, of both purple and red urchins. So maybe that's a good sign. They're still very abundant, but maybe that's not so bad. And the other point I wanted to make is that we've seen high abundance immediately before the start of the commercial urchin fishery back in 1984, and then high abundance after the marine heat wave. And we don't know anything about their abundance before 1984. This series of graphs here just shows temporal variability in the size of urchins. So small urchins on the left, large red urchins on the right, out at Orford Reef. And if we look through those and do a quasi-cohort analysis, we can go back and think, well, when did settlement occur? And then maybe back in 2011, 2012, a major episodic recruitment event, or when we saw them show up in our observations in 2014. At that point, they were probably 2 to 3 years old. And then it followed them progressing as they age through 2016, 2019. And interesting, the latest surveys in 2024 have shown that the animals have not really increased in size. So they're up there at about 10 to 12 years old, but really they're poorly nourished. So they're persisting out there, but not increasing in size, and perhaps decreasing in abundance. Okay, just to keep moving here, our grant program was looking at several different study areas for scuba dives and also shallow water or deeper water dives done with a remotely operated vehicle, and a series of study sites here in the Cape Arago area or down in the Port Orford, Cape Blanco, down off Gold Beach, and down in the southern Oregon coast, Brookings. Scuba dives done in collaboration with the University of Oregon, Oregon Institute of Marine Biology, have given us some data sets to look at the abundance, compare red and purple urchins down along the southern Oregon coast. So this series of histograms just shows their abundance in terms of density or numbers of urchins per meter squared. These are high numbers. But several points come from this by looking at this. There are higher densities and greater variability for purple urchins in comparison to red urchins, and very high densities of purple urchins at some sites like Chetco Point, Macklin Cove, Gregory Point, Lighthouse Beach down there, and moderate densities at other areas, Cape Arago, Simpson Reef, South Cove, Sunset Bay, and also out at Redfish Rocks Marine Reserve. Our group has secured grant funds from the Oregon Ocean Science Trust to conduct a whole new series of scuba dives. We did some of those this year, and we'll continue into 2025 at a whole series of sites. So we're going to continue to gather new information and build on this database. So we're looking at 20 different study sites and 50 scuba dives down there. And then with those existing funds, Scott has worked to address objective number 2, to conduct these ROV surveys in deep rocky reef sites at particular areas that are beyond the depth available to scuba divers. And for that, we use small, remotely operated Blue Robotics ROV. And Scott just finished off these surveys, so thanks for this map, Scott, showing the location for a series of 185 ROV dives that he completed this summer in these areas here over off Depot Bay, Cape Arago, Cape Blanco, down into Port Orford, at the Rogue Reef, pioneering work out at Rogue Reef, Mac Arch, and then down into Rogue Reef, or down into Brookings. And some problems, huh, Scott? So there's you troubleshooting, fixing this thing on the fly. I want to just wrap up with some pictures, just some very neat imagery. There's Scott running the show out there, the ROV in the water deployment out off the Rogue Reef. So we're looking forward to hearing. Cinnamon, please invite Scott to give a talk about this. And I want to thank Joanna Lyle, working with Scott, just some images of what we saw out there. Some very interesting sites, mixed groups of red and purple urchins in these deeper water habitats, no drift algae in the photographs, and the sea urchins appear to be actively foraging. Don't really know what they're doing, but to me, they just look like they're not stable in those locations. Okay, so let's finish off talking about abalone. And I just want to say dire straits for red abalone. We've seen significant declines at the northern limit of their biogeographic range. Red abalone are large marine snails that occur in shallow, rocky areas. They rely on drift algae for food. The biogeographic range for red abalone extends from Cape Arago, we're at the northern biogeographic extent, down into Baja, California. And the density is currently greatest along the northern California coast. We think that the range is shown in these yellow bullets there, so you can see they're present up in Oregon, but really at the northern limit. And ODFW surveys, we survey in that area from Cape Arago down to Brookings. California Department of Fish and Wildlife surveys in that area between San Francisco and Mendocino. And there's an area between where there are no surveys there. Why do we care about abalone? Abalone, everybody loves abalone. The trophy shells are important. Red abalone are the largest species of abalone in the world, and abalone grow to their greatest size at the northern limits, and therefore expected and is true. Oregon red abalone are the largest abalone in the world. So trophy hunting just for the shell, in addition to the meat, that's a popular, dangerous, and competitive sport. And we had that opportunity here. But unfortunately, our abalone have suffered a decline. And just some numbers there showing the decline at sites in Brookings and Port Orford. Back in 2015, one of our transects observed 54 abalone and roughly the same number, 53 purple sea urchins. But in 2022, 11 abalone and 11,000 purple sea urchins. And we're observing that the abalone are suffering. They're emerging out of the cracks and crevices where they would normally live to capture food. and they're migrating out, seeking food, starving, and finding empty shells. Not a good situation. Kendall Smith, ODFW shellfish biologist down in the South Coast, recently completed her master's thesis looking at the genetic makeup, the genetic signature of our red abalone populations here and comparing that to samples from California. Well, what did she find? She took tissue samples from red abalone in Oregon and analyzed them for genetic signature. The Oregon abalone exhibit a high level of genetic similarity with California. And we interpret that the red abalone in Oregon are part of a regional big metapopulation that extends from Oregon down to Baja. The Oregon subgroup is not self-sustaining and relies on periodic input of larvae, probably coming from California. Abalone naturally occur at very low densities in the northern limit of their biogeographic ranges, and we believe their densities are below the critical threshold required for successful reproduction. We're also in the situation where environmental conditions are poor with the loss of kelp and understory seaweed, not very much drift algae out there. Consequently, the Oregon Fish and Wildlife Commission suspended sport harvest in 2018 to 2024 with an indefinite closure in 2023. So we're now indefinite. And then Kendall and Scott and I have worked together to put together the conservation fisheries management plan for red abalone extending roughly from now over the next decade. There's that photograph again. One of the satisfying parts about this job is to take science, take information, and take it to the commissioners that are responsible for making the rules, put in our pitch, make a convincing argument, and come together on a decision to do the right thing, even if that means suspending a popular fishery for forever until they come back. So the factors that have contributed to the decline of abalone, these low densities result in poor reproductive success. Clustered abalone are easy targets for human harvest and also for natural harvest by octopus and other predators. There's very little food out there, hardly any kelp in some locations for foraging red abalone. The kelp beds are reduced at some sites, perhaps in response to this marine heat wave. Increase in purple urchins has constituted a competition for abalone or for food. Sea star wasting syndrome really is an absence of urchin predators. And of course, sea otters are missing, as they have been for 100 years, from our nearshore habitats. So again, just that big picture that we've seen these habitat transition from kelp forests to sea urchin dominated barrens in some locations, and not everywhere, but enough that that is concerning. So the conclusions I want to leave you with is that our rocky reef habitats and communities have experienced substantial shifts over the past decade along the Oregon coast. Changing ocean conditions, marine heat waves and ocean warming are likely the primary driver for this transition. The changes that we've seen and documented are now building that baseline characterization include declines in kelp, the mass mortality of sunflower sea stars, dramatic increase in some locations in sea urchins, and the decline of abalone. Thinking about that, these trophic cascades are the links within the food web in the kelp habitat. We recognize they're complex and they require much further study. And our team, in collaboration with the Oregon Kelp Alliance and investigators from Oregon State University, University of Oregon, the Volunteer Reef Check Program, and others, were engaged in surveys and research and monitoring to establish these new baselines. So my final thoughts for this with regard to the climate and ocean drivers: persistent marine heat waves and warming ocean waters are a regional manifestation of climate change here along the West Coast. And the shifts in ocean drivers exert multiple stressors on marine habitats and communities along the Oregon coast. Some of the casualties that we've talked about: alteration of marine and estuarine habitats. I haven't talked about the estuarine side of this, but on the marine side, seen massive loss of sea stars, increased abundance of sea urchins, mostly purple, a decline in the spatial extent of bull kelp, a decline in the abundance of abalone. On the beaches here in Oregon, we are seeing increased harmful algal blooms and increased frequency of shellfish closures, both in razor clams and crab. We're in the estuaries seeing patchy declines in eelgrass And of course, now also the increased abundance of non-indigenous European green crab. Those are just some of the ecological consequences that we've seen over this past decade. So the take-home message, my last couple slides here, I started off by saying that the magnitude of ecological change in Oregon's rocky reef habitat is unprecedented in recorded history. And is that really true? You know, do we know about that? What do we know past way in the back? We don't really. You know, we have anecdotes of how things used to be, but it's really important to actually get measurements. And so that really, it feels like our job to go on out and characterize in this quantitative way special index sites using the same types of metrics so that we can really put together that time series. And key questions then, do these current communities represent a shifting baseline that's biased by our own careers, you know, the lifespan of our careers? Is it a new normal now for Oregon's rocky reef habitats? Will these communities continue to respond and adapt into some type of alternate stable state? And how stable is it? What about resilience? Will it be possible to help enhance these suffering populations of sea stars and abalone and kelp somehow to assist recovery? At what magnitude would those type of anthropogenic enhancement activities or restorative activities need to occur to be meaningful on an ecological scale? So with that, I want to wrap it up and say thank you for the opportunity to talk about this. Kind of a downer story, but I think it's an important one, and I'm glad to be you know, helping put together this important data set. So thanks very much.
Thank you so much, Steve, for pulling that all together and telling that story. We're going to open it up to questions, and we're going to start with questions online, and I can see we've got one. So go ahead, Roseanne.
One slide suggested a comparably significant population of urchins in 1984 is today's populations. Do we know why?
The question is, why did we have such a big population in 1984?
Yes.
Yes, so we don't really know, but that's really when our program, ODFW program, started monitoring. And that was also the very beginnings of the commercial sea urchin fishery. So that was an unfished, relatively unfished, unharvested population there that had accumulated for many, many years, many decades, without major predators, sea otters. And so it could have been an unusually high overabundance there at that period, is how we interpret that.
Great. Questions in the room? Thank you.
Has there been work done on changing sand cover in the areas around kelp beds? I always thought that sand might influence the beginnings of the kelp beds for Nearshore in the spring, but I don't know much about sand cover on the bottom other than intertidal.
Yeah, thanks for that question. Gail asked about how important is sand buildup and burial and scour, perhaps? I don't know, but I would, again, in the post facto mode, say perhaps sand scour could really be a limiting factor if it's moving around and disturbing those fragile gametophytes and you know, early sporophytes. It's possible. Yes. Yes. Yep.
All right, we got another question online. Go ahead, Roseanne.
SST got a lot of attention. Could you comment on OA, calcium carbonate, and aragonite state impacts on your suite of SGCNs? And there's a PS with a lot more abbreviations here. PS, better give a shout out to OSU when talking about UO/OIMB. Scientific scuba ducks need the beeves to go diving, all in good fun.
That's great. That's really a loaded question, and it's a good question as well. The SST, of course, is sea surface temperature, and that's what I talked about, marine heat waves here. But those other important drivers, the calcium, the buildup of CO2, ocean acidification is really what they're talking about there. You know, how important could be the buildup of CO2 in seawater and the acidified ocean waters on these animals? And yes, there has been some work that has shown that early stages of sea urchin development are susceptible to disruption in terms of growth. And so that's a possibility. There are other issues associated with fertilization success that's been looked at. And the scientists here at OSU have done a great pioneering job. Not only that, I didn't hear it when you were going through that, I was listening for the H, the hypoxia situation as well. And that's the importance really of getting time series measurements right at the point where these ecological interactions are taking place. Unfortunately, we don't have those key critical measures for PCO2, the OAH parameters, and hypoxia right at the sites. Hopefully our marine reserves program will be able to build the capability to get those important time series measures so that we could really talk about those. Yep.
All right, questions in the room?
Can I add one more? Then the last part was, yes, the University of Oregon Ducks Scuba program operates as a sub-program within Oregon State University. That's true. Yes.
Thanks, Steve. That was a really great overview. And I know that echinoderms and their larvae are your original love. So even though this is a sad story, I'm happy for you and for us that you get to explore this and help understand it. And I'm curious about a couple of things. One, the sites where the kelp is persisting.
Yep.
I guess, what do you think is going on there if you're willing to extrapolate? And do you think it could have something to do with other predators? And if you took off your ODF&W hat, I guess, and put back on your larval ecology hat, is there something else that you've been seeing or that you think the baseline that we're missing that you wish we were paying attention to?
Yeah. Thanks, Jessica, for that. So sites that we saw persistence in that comparison between 2010 and 2022, that was the Rogue Reef system offshore from Gold Beach and also Cape Arago. And we also saw some unusually high abundance of bull kelp off of Depot Bay, unexpected too, popped up there. Why those locations? There's been some speculation that the proximity of the Rogue Reef system to the Rogue River may have had something to do with it. Rogue River is a pretty substantial river discharging out there, and I don't know the flow dynamics, but there's been some speculation about that. Scott has mentioned to me that the kelp bed is barely hanging on. Is that right, Scott? Declined this year. So Rogue Reef may go the same way as well. Cape Arago has been hanging in there, especially South Cove and that area between the South Cove of Cape Arago extending down the coast towards Bandon. Looks like it's okay. That's outside of any major, freshwater influence. I don't know. I really don't know. So if I could speculate and go back into that post facto mode, if I were, 20 years younger and just starting up a research program, I'd be looking at these post-larvae, the ecological dynamics in the first month of life in those kind of situations, both exposure to predators, competitors, to the availability of turf algae growing up, and a whole bunch of different things looking at that microscopic situation in the ecology of these subtitle species that really is unexplored.
All right, question, Roseanne, go ahead.
Great presentation and disturbing. Are there comparable efforts to measure changes in Oregon estuaries? And if so, how are estuaries doing relative to coastal reefs?
Yes, so excellent question. On the estuarine side, we've seen, I just alluded to them in the end, but we've seen some major changes as well. OSU professor Scott Happel has just recently published a retrospective look at the community and densities of fish, and the biodiversity of estuarine fish in Yaquina Bay has declined significantly. There's nodding head going over there. My colleagues at the South Slough National Estuarine Research Reserve, working in the Coos Estuary down in the South Central Coast, have also done the same thing, looked back at data sets from the 80s and did comparable work and have shown as well 80% decrease in the biomass and biodiversity of fishes in the estuary. That's bad news. We're seeing declines in the spatial cover of eelgrass at some sites here in Yaquina, some areas. Down in Coos Bay, some areas have really declined. It hurts me personally because I set up the monitoring program there, and as soon as I left and came up here, the eelgrass went all to heck. anyways, it's declined down there and unknown really why. And then, of course, I mentioned the increased abundance of European green crab. They've been here for 20 years or so, and they were puttering along at very low abundance until these successive prolonged warming events. And now their populations have bounded up so that they are self-perpetuating along the Oregon coast. That's some bad news.
So some good news about long-term monitoring.
Yes, long-term monitoring. I really want to put the pitch in that long-term monitoring is super important. It's not always glamorous, but it really provides valuable picture in terms of understanding what has occurred over time. So the methods, the locations, the species themselves, we really need to continue to invest in monitoring particular sites. Can't do everything everywhere, but particular priority sites, these species of greatest conservation need that really can attract funding into the state, into our program. That's why those documents are so important to be able to put together a time series story like this. Because if we just did hit and miss all over, we wouldn't really be able to tell a coherent story. Yeah, Paul.
So hang on, everybody. We are at time, and I want to be respectful of those that need to get out of here because of the weather and those kind of things. So for folks online, if you have continuing questions, Steve has put his information up on the last slide here. For folks in the room that do want to stick around, Steve, can you stick around for just a little bit? You can come up and ask questions. But before we let Steve go off of the floor and out of the spotlight, thank you so much for being a part of our community and taking this long-term monitoring so seriously.
All right. Thanks again for the invitation and for giving me the chance to just talk about this.
All right, everybody. Thank you so much. Thanks, everyone.